Myself, Mr. Mate HT, Department of Botany, RBNB College, Sri Rampur. So, today we are going to see the T.Y.B.Sc Botany, paper first, BO 331, Cryptogamic Botany, and from this paper, topic number 3, that is the life cycle of the Chara. Now, Chara is the freshwater alga. It is found in India and also all over the world. All over the world, there are about 294 species are there, and in India, there are about 69 species. This figure is according to the B.R. Vasishta. Okay, first that is the classification. Now, division is a chlorophyta. It is because of the dominant pigments are chlorophyll A, B, etc. Then, class Charophyceae, plant body is erect and have nodes and internodes. Order Charales, sex organs are complex organization and sexual reproduction is gamous type. Then family Characeae, Oogonia has 5 corona cells at the apex of the Oogonia. Then characteristic globule and nucleus are there. Genus Chara, presence of a quantication at the lower node of the main axis and nucleus present above the globule. Now, occurrence. Generally, this Chara is a freshwater alga. It is found in the freshwater reservoirs like pond, pools, ditches, etc. So, it is a freshwater alga. Now, thallus structure. The thallus structure is divided into the two parts. That is the main axis and the rhizoid. Its length is about 1 meter in length, but in generally it ranges in between 20 to 30 centimeter in height. But in some species it is 1 meter also. For example, some Indian species are Chara zhelenica, Chara hati, etc. These are the some Indian species. So we are going to study the thallus structure. फिगर करतो सर Okay, this is the thallus structure of the Chara species. It shows the different types of the branches as in the figure. For example, the main axis is getting divided into nodes and internodes. So this is the internode in between the two nodal region, and this is the node region. Okay, and this is the long branches. Okay, this is the diagrammatic representation of the thallus structure of the Chara. The plant body is getting divided into the two parts. The first, that is the rhizoid. It is important for the attachment at the substratum, and it is uniseriate and it shows the oblique septa. The oblique septa are like this. These are the oblique septa. They are uniseriate and they are important for absorption of minerals, and they are also important for the vegetative reproduction. That means they develop the primary protonema, which are important for the vegetative reproduction. Now second part, that is the main axis. So this is the main axis. It is very long. Generally it ranges in between 20 to 30 centimeter in length, but sometimes in some species it may be up to 1 meter in length also. And it is getting divided into nodes and internodes. And the lower nodal region of this main axis shows the quantication. Now this main axis shows the 4 different types of the branches. From this, number 1, that is the long branch. So these are the long branch, and they are also called as the branches of unlimited growth. And the structure of long branches is the same as the main axis. That means that these long branches, they are also get divided into nodes and internodes. Now this is the first. Second, that is the primary laterals. They are also called as the branches of limited growth because they are developed in a whorl, and in each whorl there are about 60 to 20 primary laterals are getting developed. These branches also show the nodes and internodes. They show up to 15 nodes. So this is the second branch, which is called as the primary lateral branch. Now there is another type of a branch which is developed on the nodal region of the primary lateral. These branches are called as the secondary laterals. They are also developed in whorl on the nodal region of the primary lateral, and they are single celled. They are also called as the leaves. So these are the 3 different types of the branches, that is a long branch, primary lateral and secondary lateral. So the 4th type of the branch, that is the Stipulator. So this is the 4th type of the branch which is developed on the thallus of the Chara. Now this is the primary lateral branch. This is the primary lateral branch, and This is the secondary lateral branch, and in the axial of the secondary lateral branch, there a small lip-like structure is developed. That structure is called as the stipulode. When the number of stipulode is the same as the secondary lateral, then such a type of species are called as such a type of species of Chara are called as the unistipulate or haplostipulate. For example, Chara brownie. And when the number of stipulode is more, it is double than the secondary lateral, then such a type of the species of the Chara are called as the bistipulate or diplostipulate. For example, Chara nuda. So this is about the stipulode. Now next, that is the cortication. The cortication is generally seen on the lower internodal region of the main axis. Now there are some filaments. The filaments which develop from the upper node and goes toward the lower side, such type of filaments are called as the descending filaments. And some filaments which develop from a lower node and goes toward the upper side, they are called as the ascending filament. And both of these type ascending and descending filament, they meet at the center. And this structure is called as the cortication. Generally, it is seen on the lower internodal region of the main axis only. Now, cell structure. Now this is the cell structure of the internode of the thallus of the Chara. Now it is somewhat rectangular in shape. The outermost layer is called as the cell wall, and generally it is synthesized from the cellulose. Inside this there is the cytoplasm, and at the center there is the central vacuole, and there are a number of nuclei are found in each cell, and there is a discoid type of a chloroplast is developed in the Chara cell. Thank you.